Okay, so good morning everybody. We're back to the seminars. Today we have two PhD students that will present their PhD plan, their thesis, and we will start with Lucía, who is originally from Madrid, where she studied biology, and then she moved to Seville to do the master in Biodiversity and Conservation or something like that, from the UPO, from the Pablo de Olavide University, and she started to work here with Godoy to do her thesis. So that's all. Yeah, we'll have 20 minutes for each presentation, followed by 10 minutes of questions, okay? So that's all from my part. Lucía, the floor is yours.
Well, hello everyone, I'm Lucía, and I'm a second-year PhD student working in José Antonio Godoy's group, and today I'm going to be presenting my PhD plan, which is titled The Genomic Basis of Inbreeding Depression and Deleterious Traits in the Iberian Lynx. So, as you may already know, the Iberian lynx was once considered the most threatened feline in the world, and is still categorized as endangered by the IUCN. This was mainly caused by the population decline that the species has been suffering, as it has been the subject of various severe bottlenecks, the last one occurring in the last century. In this map, we can see the distribution range of the species throughout the last century, and we can observe that by 2002, there were only approximately 100 individuals, and they were divided into small and isolated populations. One was found in the Doñana area with approximately 40 individuals, and the other one in Sierra Morena in Andújar with more or less 60 individuals. This fragmentation and isolation led to concerning levels of genetic erosion in the species, which were manifested by its high inbreeding coefficients, the high differentiation between the two populations, as we can observe in this structure plot over here, and in the high FST value of 0.22. And additionally, it also had very low levels of genetic diversity, shown in this plot of Abascal and colleagues, where they are comparing the number of heterozygous SNPs per megabase in the Iberian lynx and other species. And we can see that it has even lower genetic diversity levels than a domestic inbred cat. This accumulation of inbreeding and the loss of genetic diversity can lead to a fitness reduction. And in the Iberian lynx, there are several evidences of this fitness reduction. We have a smaller litter size, a reduced immunocompetence, a lower quality semen, which is also correlated with lower levels of heterozygosity. And additionally, several deleterious traits with a presumed genetic origin have been detected in the captive population. So with all this unfavorable scenario, a series of conservation actions were established. Among those measures, we can find a captive breeding program, which took founders from the two remnant populations. Also, individuals were translocated from Sierra Morena to Doñana, and captive-born individuals are being reintroduced in areas that were previously occupied by the species. And we have three main large reintroduction areas. One in Montes de Toledo, another one in Extremadura, and another one in Vale do Guadiana in Portugal. All of this was possible thanks to many different conservation projects, throughout which a lot of samples have been collected and a lot of information has been gathered. So the main goal of our current project in our group is to take an advantage on the availability of samples from all these different populations, on data that we have on demographics, phenotypes, and genealogies, and on the availability of a new high-quality reference genome, which is chromosome-level annotated. And specifically, during my PhD, I want to use genomic tools to understand the consequences of the population decline on functional genomic variation, in order to try to discover what are the mechanisms underlying inbreeding depression, which has so far being hindered by the limitations of other traditional genetic markers in the study of adaptive genetic variation. So to do this, we have decided to divide the PhD into 3 chapters. In the first one, we want to identify regions in the genome that are associated with already diagnosed genetic diseases. In the second one, we want to estimate global inbreeding coefficients and try to characterize these inbreeding local patterns in the genome. And we are also going to try to detect candidate regions with a lethal effect. And in the last one, we want to identify genomic regions that are associated with this fitness reduction. And to do all of this, we need to generate new sequencing data. And we wanted to do so for a representative proportion of the Iberian lynx population. So for that, we decided to follow the next strategy. First of all, we have already sequenced 50 individuals at a high coverage of around 30x, which will serve as a reference panel for the imputation of up to 300 individuals more sequenced at very low coverage of around 1x. This phasing and imputation consists of trying to fill the regions in the genome where, with a lot of missing genotypes that result from low coverage sequencing by using the information of haplotypes that already exist in our population and that are collected in this reference panel here. Like that, we can obtain these missing genotypes to obtain whole genome haplotype data. And we hope that this imputation strategy is quite accurate in our species because of the high linkage disequilibrium and the high degree of relatedness within the species. But in any case, we can always try to check to validate the imputed haplotypes by looking for Mendelian inconsistencies thanks to the availability of pedigree in all the populations. And so how did we choose these 50 individuals? We wanted to capture the maximum diversity of haplotypes in our population to improve the imputation accuracy. So we decided to select the individuals that had a higher number of descendants and that are minimally related between them. And we chose individuals that were founders from the 2 remnant populations, individuals from Doñana before and after the translocations, because we are also interested in the study of genetic rescue in the species. And we also sequenced translocated individuals and reintroduced ones. Once the sample were selected, we extracted the DNA here in the molecular ecology lab and sent it to be sequenced. And when we received the data, we performed a quite standard bioinformatic analysis, which consisted on the raw reads, quality control, trimming, alignment, and the variant calling, filtering, and phasing. Phasing, in case you're not familiar with the concept, just means trying to determine which chromosome each allele is coming from in order to define these haplotypes. And I've just recently finished with this process, obtaining a reference panel of 50 individuals with an average depth of coverage of 26 and with 1.33 million SNPs. So this would be ready to be used for imputation for the low coverage sequencing data. For this data, we have already sequenced 75 individuals that are coming from the captive populations, and they include 20 calves that were diagnosed with juvenile idiopathic epilepsy, which I will talk to you more about in a few slides, and their relatives. And well, here you can see a picture of the pedigree, where in orange are the epileptic individuals, and in yellow, the yellow circles represent the individuals that we have already sequenced, either at high or low coverage. And apart from this, we are also preparing another set of individuals from Doñana, from before and after the translocations, minimally related, and that have some fitness data available. And again, we extracted the DNA. In this case, we are preparing the libraries ourselves in the lab, and we send them to sequence, and another bioinformatic analysis, quite similar to the previous one. So for the set of 75 individuals, we have obtained a coverage of 1x, which is more or less what we were targeting. And so this is the stage that I'm currently working on testing how the imputation works, testing the softwares. So I cannot give you any results yet, but this is our next step. And once this is done, we can start with the interesting analysis. So in the first chapter, as I mentioned before, we're going to try to identify regions in the genome that are associated with already diagnosed genetic diseases. We are mainly going to focus on the study of juvenile idiopathic epilepsy, which was diagnosed in 20 cubs in the captive population. Because even though the captive management has maintained adequate levels of inbreeding, as these captive populations are usually founded by a small number of individuals, the risk of recessive deleterious variants becoming expressed in homozygosity increases, and new traits can appear, new detrimental traits can appear in these populations, which is the case of epilepsy. We have several evidences that it has a genetic origin. First of all, when the disease was diagnosed, they changed the mating system, assuming a recessive mode of inheritance, and no more new cases have been diagnosed since 2018. So it seems that that change was quite successful. Additionally, these epileptic individuals have higher inbreeding and lower hybridity levels than unaffected ones. And different pedigree studies have estimated that it has a high heritability, very high, of 0.97, that it has a recessive mode of inheritance, and that is governed by one or two major effect genes. So the main goal of this study would be to try to unveil what are the genetic bases of epilepsy in order to identify variants that can help us to identify the true carriers of the disease, so we can incorporate this information into the genetic management of the species. So to do this, we know that our sample size is quite small, so we are trying to use information from two different approaches. In the first one, we want to use information from the pedigree, because knowing how individuals are related, you can do different type of analysis. For example, if you have information from the parent and the affected offspring, you can do a transmission disequilibrium test, which consists on searching for deviations in the expected Mendelian inheritance. If you have information from the whole families, you can also do a linkage mapping, which looks if some alleles are preferentially transmitted to affected individuals than to unaffected ones. And also with siblings, we would like to do some what is called homozygosity mapping, which consists on searching for homozygosity tracts with shared alleles in the affected individuals and that are absent in the unaffected ones. And apart from that, we also want to perform genome-wide association analysis, which uses information from two groups of individuals that must be unrelated. And one group would be the affected ones, the other one the healthy ones, and you just try to compare and look for differences in the allelic frequencies. And once these analyses are done, we hope to get some candidate regions that would need to be further explored. So we would like to see if there are some genes annotated there, if there are some deleterious variants, because I haven't mentioned this before, but we are also going to try to perform some functional annotation of the SNPs, which consists of assigning them a category depending on their predicted level of deleteriousness. And we want to try to identify the causal variant. This is what we refer to with fine mapping. Because normally when you do a GWAS, you get a peak, you get a lot of SNPs in the same region that show a significant signal, but then that's because they are already linked, they are all linked to the causal variant. So fine mapping consists of trying to identify which is the more plausible causative variant. And for that, we can look at the level of deleteriousness, we can try to find if there's some structural variation there, or we could even do some targeted sequencing of the regions of interest. And that would be the first chapter. And in the second one, we want to characterize these patterns of inbreeding depression in the genome, both globally and locally, and see if there are some regions with a lethal effect. So to do that, we are mainly going to focus on the study of runs of homozygosity, which are contiguous regions in the genome where all the loci appear in homozygosis, and they arise from inbred crosses, because an individual is inheriting identical copies of the same haplotypes because they are coming all from the same ancestor. In this case, they are represented here in blue, and you see that they are all coming from this grandparent. And once these rows are defined, we can calculate the F row, which is an inbreeding coefficient that reflects the proportion of the genome that is found in these regions. And it's a quite precise estimation of inbreeding. It's not like the inbreeding coefficients you expect to get from the pedigree. And also with the rows definition, you can study their length, because the longer the rows are, the more recent the inbreeding event was, as they are broken down over generations during meiosis because of recombination. We also want to see if there are some regions that are common in all of the individuals that always appear in row or that never appear in these rows. This is what we refer to with deserts and islands. We also want to study their correlation with the demographic history to perform a functional analysis to see what's inside of them and to see if they are enriched in deleterious variants. So as you can see, there are many possibilities here. And in the second part of this chapter, we want to try to identify regions that have a potential lethal effect. The idea here would be to look for alleles, recessive alleles, that are so deleterious that when they appear in homozygosity, they cause the death of the individual. So, as we are targeting embryonic effect, this means that homozygous individuals would never be born, and they would therefore never be sequenced. So as a consequence, you can use population genomic information to try to look for chromosomal regions that are common in the population, haplotypes that are common in the population, but that they never appear in homozygosis. This is what I try to represent here. So for example, in red would be the deleterious allele, and this would be an inbred cross. So you can see that when the two haplotypes appear in homozygosity, this individual would never be born. So you will always find this combination of haplotypes in the population. Even if it sounds a little bit weird, this is an analysis that has proven to be useful in many livestock species, but it's true that it requires quite large sample sizes. But we are hoping to get there. And again, this would only give us a list of potential candidates, regions. Then we need to further explore what they contain. And in the last chapter, we also want to find regions in the genome that are associated to a fitness reduction in the individuals. So for that, we want to first study if there's a correlation between the global inbreeding levels and the fitness reduction. So how can we obtain these fitness estimates? In the case of the captive population, it's quite straightforward because it has been very intensively monitored. So we have data on health, on reproduction, on physiology, and the perinatal survival, etc. And for the wild population, it's a little bit more complicated, but we are hoping to get some variables from capture-mark recapture models, like the apparent survival rate or the annual reproductive success. And apart from that, there is also a very valuable source of information, which is the pedigree. In the captive population, it's quite precise and extensive. And in the wild ones, it can be reconstructed. For example, a master's student in our group, Kaya Mora, reconstructed the pedigree of Doñana. We have an updated version by using 300 SNPs that are coming from non-invasive samples. and we hope to do the same with the reintroduced populations also. And from this pedigree, we can obtain variables like the lifetime reproductive success, the litter size, or the per capita recruitment rate. And once this global effect and this correlation is proven, we would like to, again, find regions in the genome that are responsible for this fitness reduction, also through genome-wide, different types of genome-wide association analysis. And that would be all. I would like to thank everyone in my research group for being a daily source of motivation and inspiration, and thank you all for listening.
Okay, so any questions from the audience? Just to remember, only constructive questions, because they are PhD students. Maybe some of you might want to move there. Okay, Xavi.
Yeah, thank you, Lucía, for your presentation. So you said that you expect to get 1.3 million high-quality SNPs from your genomic analysis?
That's what we have obtained with the
All
50
right.
samples sequenced at high coverage.
All right. So do you have an estimate of the proportion of genes with informative SNPs in it?
No, I haven't really looked at that yet. Sorry, we haven't performed a deeper analysis with that.
Okay, well, it's not the same whether you have SNPs in promoters, introns, extrons,
Yeah, for sure.
regions,
That will be done. The next step is to do the functional annotation of the SNPs. So that can also give us some hints if they are found in introns, in exons, if they are synonymous or non-synonymous, which is the level of deleteriousness. Okay. So we hope to do that next.
Maybe I don't need it.
No, it's for the people in YouTube.
So you mentioned a change in the mating system. Did you mean the captive program?
Yeah, in the captive program.
Can you give us more details about that? So that was intended, I mean, I guess it was intended, so it was kind of a design or...
Is, of course, they didn't breed the individuals that were diagnosed with epilepsy, and they tried to avoid the mating between the potential carriers of the deleterious alleles. So if two parents have had an offspring that had epilepsy, they didn't mate them again, and they didn't mate them with other parents of other epileptic calves. Assuming that they have an heterozygosity, the deleterious allele is heterozygous in there, so they have the allele, but it's not expressed. So if you mate them again, you can get more homozygous offspring and then more epileptic individuals.
If you don't know if they have the disease, is there a chance maybe changing the mating system for females maybe to be polyandrous, to sample different males? Maybe,
can you repeat the question?
Yeah, so is there a chance you can maybe know whether females mating polyandrously that will avoid this disease to spread? Maybe if the females sample different males, even if you don't know whether they have the disease.
The females?
Sorry, yeah, I mean in the field, do they mate with multiple males, the females maybe, or? I think
it's the matings change. You know, the partners change.
Yeah.
They never keep one individual with the same one throughout all their lives. What they do when they detect epilepsy is try not to mate them with another individual that had epileptic descendants. Because at that time there were not so many founders in the population that you could just say, they had an epileptic offspring, let's not breed them again.
No, I see that, but you need to know whether they have the disease or not, the potential partners. Yeah. So my question is, if you don't know whether they have the disease because you have maybe a large sample of individuals, whether mating them other than monogamously would prevent the disease to spread in the population.
Supposedly.
So in other words, I'm wondering about, it's important to have in mind the mating system for the conservation of the species.
Yeah, I think
in nature.
For sure.
Okay.
I don't know if I have answered your question.
Yeah, sure, totally. Yeah.
Okay.
Thank you.
In your third chapter about comparing fitness between captive and population in the wild, have you thought about a way of taking into account differences in the environment?
Yeah, in this first chapter is still yet to be defined, but I don't think that it's appropriate to mix populations. Like the captive population has very specific conditions, and the type of analysis that you can, the type of fitness data that you can get is probably very different from the captive populations than from the wild ones. So we are not very sure yet how we will do this analysis, but for sure environmental variables will need to be taken into account.
You have some individuals that were in the wild, and then you keep them in
the founders of the captive populations were born in the wild, and then they are in the captive population. But still, we need to define which specific variables will be used. We don't know yet, but thank you.
Well, people in YouTube can ask as well. I think that there is no. Okay. Okay, one more. Last one.
Thank you, Lucía, for the presentation. So in chapter 2, you talk about the sample size required. Do you have an estimate of the which can be the critical sample size that you need to estimate robustly the lethality frequencies? And if you don't get that sample size, is there a B plan for that?
I think here the sample size also depends on how deep you want to get in the analysis, because with a small sample size, the variance at lower frequency will be impossible to detect. So in livestock species, which are the ones that this analysis is mainly done with, they are talking about thousands of individuals, but for example, there was a study that is done with only 150 horses, and there what they are detecting is variants that are at quite high frequencies in the population, but that they still are lethal, because there's an inbred population with a very small effective size. So we don't have an estimate of the adequate sample size. But we hope that we will try to sequence as much as we can and see if we can get something from there.
No more? Okay, so thank you very much, Lucía. Anyway. If there are more questions, you can always ask after the talk. And now I'm going to introduce Iago. Iago is originally from Galicia, but he also moved to Seville to do the UPO master as Lucía, the Biodiversity and Conservation. But in this case, he's doing his thesis between here in the EBD and the Museo with Ana Benítez, no, there in Eloy and Pedro Jordano. That's it from my side.
Thank you, Javier, for introducing me on this talk, in which I pretend to talk a little bit about my PhD. As Javier said, my supervisors are Ana Benítez López and Eloy Revilla, and as well Pedro Jordano. And my PhD is focused mainly in one concept, driven by one human activity, that is overhunting. And I would like to introduce the talk by talking a little bit about the concept of defaunation that refers to the loss of animal species mostly, but as well to the local abundance declines. And to these loss of animal species, we have different drivers. The two most important ones are habitat loss or habitat modification, and potentially the second one and most widespread in tropical ecosystems is overhunting. And one particular feature of hunting-induced defaunation is that hunters, since it tends to target larger animals and larger species, since it renders a larger quantity of meat, we have that through their impacts, we have a downsized communities in body size. And when it comes as well to tropical ecosystems, we found that hunters tend to act following the central foraging hypothesis that states that hunting pressure decays as the accessibility of hunters to a given site decreases. This is that, for example, one human settlement, one road, or one river, we have it that hunting occurs mostly nearby these settlements. So this creates some kind of gradients where we have some forests that are almost completely devoid of animal species and other mosaic of areas that are completely intact in terms of overhunting impacts. Here I show you one study carried out by my supervisor, Ana Benitez, in 2017, where she estimated through a meta-analysis approach that up to 60% of bird declines occur between 0 and 7 kilometers, while for mammal species, up to 80% of mammal declines occur between 0 and 40 kilometers. But not all mammals or all bird species, in general game vertebrates, suffer these declines in biomass and abundance. And for example, we have that for some functional groups, such as game species or arboreal frugivorous or graziers, we have a local declines in abundance, but for example, other functional groups of vertebrates even increase in abundance, potentially due to ecological release of competition and predation, when their predators are extirpated, obviously. This, when we try to look at the functional roles of vertebrates, we can find that different parts of the functional space composed by all the functions of the vertebrate communities can have different risk of extinction due to human activities. Here I just use this example that, for example, we could expect that some parts of this functional space can be under a high risk of extinction due to overhunting when we extirpate these functional groups. For tropical forests, why this is important in terms of plant species? Well, both vertebrates functional space and plant functional space are connected through ecological interactions. Here in this talk, I will focus mostly in mutualistic interactions that drive two processes that are seed dispersal and seed predation, that in ultimate terms, it mediates also the forest regeneration. Here, for example, I show you one conceptual framework of one ecological network of fruit and frugivorous interactions, and how, for example, different species can interact with different subsets of the functional space of plant species across tropical forests. For example, we have that in function of body size, we could expect that for a trait matching, only small body size frugivorous could only interact with one part of the functional space of plant species, while, for example, larger frugivorous can interact with a wider range of plants. This comes also with the functional importance of large body size frugivorous when we talk about seed dispersal. And this is to three main points. That would be that large frugivorous are able to swallow larger seeds compared to small frugivorous. Also, they offer longer good retention times that is also translated to a higher capacity to move seeds far away from mother plants and escape Janssen-Colnell effects. And also they have, in general, high energy requirements, so they need to consume a wider amount of fruits and seeds. In tropical systems, we have also another relationship with larger fruits. That is one trait of this functional space of good plant species, that is good density. So here I show you some results from Carolina Bello in these papers, where she found that the longer the seed diameter, the longer the good density of plant species. And since carbon storage can scale with good density, we could expect that the non-random functional extinction of larger frugivorous might hinder the limit of carbon stockpiling in tropical forests. So through these cascading effects of overhunting, we could expect that through an initial community with all potential vertebrates occurring there, we have that over these overhunting impacts, we have a directed and non-random extinction of only one subset of all vertebrate community that leads to a faunally degraded or defaunated community. And why this is important, or estimated to be important, is because it is estimated that a huge percentage of woody plant species in the neotropic around the 90 and Afrotropics around the 80% of these plant species depend on these frugivorous to disseminate their seeds. And here I show you only the conceptual framework of my thesis to sum up all the things that I said. So my thesis would be to study these connections between the faunation gradients, declines in functional diversity, how this is translated into degraded ecological networks between frugivorous and plant species, and how this is ultimately related to forest regeneration and ecosystem functioning related to carbon stockpiling. Here I show you the structure or the potential objectives of my PhD thesis in 4 different chapters. I focus in 2 different spatial scales, one at pantropical scale and another at local scale. In the first chapter, my main aim is to provide a predictive outcome, or where the main hotspots of hunting-induced defaunation impacts on both bird and mammal populations are located at pantropical scales. With these predictions, I pretend to estimate losses of or changes in vertebrate biomass due to hunting, and then to estimate these functional diversity declines and overlap it with carbon stock maps globally to locate those areas that maybe we need to employ some conservation effort to avoid the long-term declines in carbon stocks through the extirpation of large frugivorous. And at local scale, I have a 4th chapter to study the impacts of overhunting in the Western Brazilian Amazon in the mutualistic frugivorous interactions, and to study how the structure of and properties of these ecological networks changes along a defaunation gradient. So just to introduce you the first chapter, I would like to emphasize in one particular feature of hunting-induced defaunation. When we talk about the drivers of defaunation, such as deforestation or wildfires that mediate habitat loss, we found that through remote sensing approaches, we are able to track them, while overhunting can be hiding under these apparently under-disturbed canopies. So remote sensing approaches are not the best ones to track hunting activities and their subsequent impacts on animal communities. So to this end, what I did was to carry out a systematical literature review where I compiled studies across all the tropics that reported abundance estimates in both hunted and non-hunted sites. And I expressed these changes in abundance due to hunting using response ratios by dividing the abundance of a given species in a hunted site by its respective abundance in the control sites. And also I extracted a set of proxies of hunting pressure from each study and each site, and that inform us how much hunting pressure is affecting these abundance estimates. How much hunting pressure is affecting these abundance estimates. We have that for example, I extracted the distance to hunter access points following the central central foraging place hypothesis that I stated previously. Also, the travel time to major cities as a proxy of proximity to urban markets. Also, the prevalence of stunting that is related with the poverty, since this predictor is related to wild meat consumption in tropical countries. Also, human population density is related to wild meat demand. Also, net primary productivity under the hypothesis that highly productive sites would have a higher level of standing game biomass. And also if hunting occurs within or without the boundaries of protected areas, since within the boundaries hunting can be banned. And hence the hunting impacts would be lower. With all of this information, I employed a Bayesian hardware model approach to model two phenomena at the same time. One is the probability of local extirpation due to hunting by accommodating a binomial distribution and modeling zero and non-zero, I mean extirpation basically. And then when it's no zero, there is an animal there, by accommodating a log normal distribution, I am able to model the changes in abundance due to hunting. So basically, if a logarithm of the response ratio is above zero, means an increase in abundance, equal to zero, no changes in abundance, and below zero would mean a decline. So basically, in these models, I incorporated all the response rating of both mammals and bird species, and I related them to a set of species traits, mainly body mass, but also other like trophic level, and drivers of hunting pressure that I mentioned previously, and two random intercepts. One is country to account for example, difference in hunting legislation in between or among tropical countries, and also one random intercept of species to account for the lack of independence, but also with a phylogenetic covariance matrix to account for the relatedness in phylogenetic terms among pair of species in my models. And with these models, what I did was to predict hunting impacts for each of all tropical bird and mammal species. And with this prediction, what I did is to aggregate into maps and calculate a defaunation index that ranged from zero, that would depict a faunally intact pixel, towards one that would mean a fully defaunated pixel. Here I show you the preliminary maps of my results that are in minor review in diversity and distribution, where we are able to map these hotspots of avian defaunation. Just to sum up, what we found is that there is different spatial patterns in function of body size for different body size classes. For example, we have that the main hotspot of defaunation for bird species is the Indo-Malayan realm, also for large body size species and medium body size species. But when it comes to large body size species, we found that, for example, we have larger amount of defaunation in both the Afrotropics and Neotropics. Here I show you the maps published by my advisor following a similar methodological approach for mammal species, and we found similar patterns where in function of the body size of a species we can found different patterns of hunting-induced defaunation, and as well the area covered by these hotspots is larger for large body size species compared to smaller ones. Okay. Right now I am at this point in my PhD thesis where I pretend to use Lucas-Santini models to predict density estimates across the distribution range of all tropical bird and mammal species. And I show you here one example used to better get the overall picture of this chapter, where I pretend to predict the density across the distribution range of all bird and mammal species under natural conditions, I mean with no hunting. With this, with information of the body mass, what I pretend is to generate maps of predicted biomass for each species under these natural conditions. But in another scenario, what I pretend is to apply my models of hunting impacts to estimate the changes in density due to this hunting. That can be one increase or one decline in function of the functional groups and the body size mostly. And with this, what I pretend is to estimate the difference in the predicted biomass in both under natural conditions and with the hunting scenario to trying to estimate how many biomass are we losing of vertebrate species across tropical forests due to hunting. In a third chapter, what I will use is these biomass changes to estimate the declines or increases in functional diversity using a set of vertebrate traits from Elton traits, Abonnet, for weird species, and quantify the changes or losses of this functional space that I talked to you previously in the previous slide. And with these changes, I pretend also to map it so we can pinpoint those areas where through hunting we are losing functional diversity of vertebrate communities. With these maps of functional diversity that I will estimate, I will overlap them with global maps of terrestrial above and below ground carbon density maps to create bivariate maps that indicate those areas that, for example, can have a high functional diversity of vertebrates while holding, for example, high density values of carbon. So there is higher levels of carbon stocks there, for example, to implement conservation measures there, but also to pinpoint where the hunting impacts are threatening through declines in functional diversity those sites that hold large values of carbon stocks or carbon density. And this would be the conceptual framework of the macroecological part of my PhD thesis. Then at a local scale, what I am doing is to record fruit-frugivore interactions in the western and Brazilian Amazon. Here I show you the location of the Medio Juruá region. I work along one defaunation gradients that irradiates from Carauari city toward two reserves. Here I show you in different colors hunting pressure and in the size of each circle the vertebrate biomass, and as you can see there is a notable difference between sites in function of hunting pressure in terms of vertebrate biomass. So for example, we could expect that in these circles we have entire vertebrate communities with a larger density of vertebrates, while when we travel along these defaunation gradients we have degraded communities. Here I show you a theoretically mutualistic interaction networks when we found that in the control side we would expect that ecological networks would show a complex structure with higher connectance and with all modules present, while we advance in these defaunation gradients we would expect that the links between both vertebrates and that are over-harvested would be lower or weaker or even disappearing in the worst of the cases. So I am studying or recording these fruit-frugivore interactions in 5 different genus that has different fruits and seed size. That is Euterpe, Pouroma, Virola, Irianthera, and Puteria. I ordered it by fruits and seed size, so it ranges from 1 centimeter to 4 centimeters, the largest one. And here, basically, I am showing how I collect the data. I am installing both camera traps in the ground and on the canopy. And I carry out already 2 fieldwork campaigns in the western Brazilian Amazon. The first one was in 2022 and 2023. It was a pilot study to test the methodology where I compiled more than 50,000 videos. I installed more than 100 camera install. We are processing the videos yet because it's a large amount, and we saw only the 40% of this first fieldwork campaign, and we recorded already almost 2,000 visits to focal plants. In the second fieldwork campaign, we installed a larger amount of camera traps stationed both in the canopy and on the ground, and I recorded more than 200,000 videos. And I still have to come back to the Medio Juruá region to carry out another fieldwork campaign in between November of 2024 and March 2025 in the wet season when most of these plant species are fruiting. With these ecological networks, what I pretend is to study how some properties, some structural properties changes along these defaunation gradients. I will only refer here to potentially the 2 most important ones, that is nestedness and modularity. For For example, modularity is, normally there is papers talking about how a modular structure of ecological networks is able to avoid the propagation of perturbation through the ecological network, since different animals interact with only a subset of plant species and give this modular structure. So we would expect that these metrics would inform us through the decline that hunting pressure is being affecting the structure of these mutualistic interactions through the extirpation of mostly the large body size frugivorous. And this should be the summary of my PhD plan. Thank you for being here listening, also to my supervisor and all the collaborators of this huge project. And that is all.
Okay. Any questions from the room?
Thank you. Thank you very much. I can think of two other means that you can have a reduction in biomass and change in abundances that are not only the faunation, the hunting of animals. One would be landscape perturbation, landscape reduction, habitat modification, and the other would be a practice that, at least in my country, happens often, is that not big animals but small animals are taken out from the forests for selling, for species trafficking, which don't target the big ones but the small ones. How would you incorporate that kind of variables that are also impacting your data but not through the faunation?
Okay. Regarding to the first question, I think that this is a strong point of this analysis, is when we extract, let me show you all this information, we have some inclusion criteria of the studies. I mean, I do not include all abundance estimates of all sites. And for example, I do not take those data from sites that has, for example, land use changes, deforestation and other drivers that can affect species abundance, for example, also habitat difference. So I am trying to isolate only the hunting impacts of vertebrate abundance. And the second one, I mean, I didn't have to have enough time to talk a lot about this, but I incorporate different traits of species, mostly body size, in function of these drivers of hunting pressure. So for example, what we found as well is that we found an increase of abundance of small mammals when the distance of the hunter axis are the lowest. So we are able even to map this ecological release. And for example, for the pet trait that you mentioned previously, I did this paper and I also plotted the impacts of the pet trait on bird species. And what we found is that with these models, I was able to differentiate two scenarios. One that is the hunting for subsistence with this structure of this model, and then one commercial scenario where most of these small body size species are hunted for the pet trait. So I don't know if I answered you totally the question.
Thank you very much.
Any more? I have one. I guess that the article is already minor revision, so it's already there. But have you considered including some life history traits? Maybe that I guess that it really affects how like the animals are affected by hunting.
Yeah, I consider, but I consider it. But here, think about that I am trying to model the hunting impacts on species abundance of all tropical bird species and mammal species, setting different predictors with covariance, and so you have some kind of trade-off to model all this stuff. for a species trait, the most important one is body size. That sum up or is correlated even with low reproductive rates and these kind of things. Yeah, I consider it, but the best modeling framework was to include, for example, trophic level or body mass and this kind of thing. But yeah, I consider it.
Well, any more questions? I have another one in case. And regarding the fourth chapter, like the gradient that you have of hunting pressure, does the forest like functional space differ between the gradient of hunting? Like, I guess that you.
I mean,
like, how do you map the vegetation? Like the functional space of the vegetation. Do you have enough species, I guess? How resilient are the networks?
Well, this chapter is based on a previous PhD thesis that was carried out by a Brazilian PhD student, Andressa Scavin, and she was totally crazy because here along this deformation gradient that is almost to 600 kilometers of river. She took the vegetation composition of plots of, I don't know, 0.1 hectares. And we have data of the abundance of both vertebrates and plant species. And I intend to do this in the future if I talk with them, because I would like to estimate the local declines in functional diversity along this deformation gradient. It's one potential chapter that I would like to include, but I have to discuss this with them.
Okay. So cool. Any more from the audience? Okay, so I guess that's all. Thanks all for coming. You can ask, like, if you have more questions, you can let them know afterwards. See you in May.